Okay.
All right, we can get started, I guess. Good evening and welcome to the Musella Foundation's May Brain Tumor Awareness Month webinar series. To kick off this series, our special guest speaker tonight is Dr. Timothy Cloughesy. Dr. Cloughesy is a renowned figure in neuro-oncology. He's the director of UCLA's neuro-oncology program, co-director of the UCLA Brain Tumor Center, and the global principal investigator for the GBM AGILE Trial. We're honored to have him present for us tonight. Dr. Cloughesy, I'll hand it over to you.
Thank you, Vanessa. And Al, thank you for inviting me to speak today. For all of you that are watching, thanks for joining. I hope to give you some insight into clinical research and in particular, how that functions with this late stage registration trial that is called GBM AGILE, which is an adaptive platform trial, which I'll get into a little bit. So first, clinical research in general, the goal is to be able to provide valuable, a valuable understanding of human disease, the idea that it could be about preventing or treating illness or promoting health. And, we're focusing more on the therapeutic interventions associated with that and the clinical trials related to drugs. And in particular, with the goal of trying to get, some of these drugs, to identify that there is evidence of benefit. And then once that is done and in a well-conducted and designed trial, that would then be able to go forward for a market application. You guys, I think, are familiar with this, but I'll just go through it. This is the typical path that we go through to get regulatory approval. This starts with kind of preclinical research, which is a mixture of identifying that a target exists, then attempting to drug that target, and then identifying a series of drugs that might be helpful in picking a clinical candidate. And once that clinical candidate is selected, there's a number of evaluations that have to go forward before it goes into patients. Those evaluations include evaluations in animals, and then a decision is made about what dose one can start with in a phase 1 setting. And the phase 1 setting looks at a series of starting with small doses and a series of cohorts that use higher and higher doses or doses that get adjusted with different cohorts based upon the presence of toxicity in some way. And in a sense, we're looking for that nice balance between what is tolerable and what seems to be providing enough drug to allow there to be some impact. And in the end, in the phase 1 setting, you come up with something called a recommended phase 2 dose. It's getting a little more complicated now with the FDA, with Project Optimus. And sometimes it's not just coming up with this dose that you think is one that is tolerable, but sometimes looking at a couple of doses and trying to understand whether there's greater tolerability with one of the 2 doses and maybe even greater efficacy with one of the 2 doses. The phase 2 and phase 3, and I will say this, during this phase 1 time, what usually happens is there's kind of a dose expansion. So you add 3 patients at a time. If you have typically 1 dose-limiting toxicity of those 3, you add an additional 3. And if you only have less than 2 of these dose-limiting toxicities, that means you could advance to the next cohort. And in the end, what happens is you run up against a ceiling of toxicity and you back down. And that's how you define your dose. And that sometimes is the recommended phase 2 dose. But then there's an expansion cohort. And many times what we do with this expansion cohort, it's about 10 or 20 patients. And we're trying to see if there's some benefit that exists there. It's actually really hard to see that benefit in that setting because it's a single-arm study. It's hard to know what you're comparing it to. It's hard to know what kind of patients were enrolled, relatively good or relatively not so good patient and their functional abilities and their expected outcomes. So that makes it unclear many times. And so that's why there can be a phase 2 setting. And what you're going to hear is that we're really combining this phase 2 setting and this phase 3 setting into GBM Agile. But what usually happens is in a phase 2 setting, you'll either do a single arm study or a randomized study, again, to try to identify some early evidence of benefit. And then if that benefit is seen, then you move forward into the phase 3 setting. And again, that's the idea here, is that GBM Agile is combining these two components here. And if you're successful then in this setting, in what the FDA calls a well-conducted and randomized trial, then that's when you move on to the possibility for market approval. So lots of risks and benefits of participating in trials. You can imagine in early phase trials, you might get too high a dose and you might get too low a dose. Certainly in the phase 3 trials or in randomized trials, that a little bit has been worked out. So it's not so much about the tolerability, and the other risks might be that you're going to get on a placebo instead of a treatment arm. But, the real benefits here, you contribute to the advancement of medicine. There's early access to experimental therapies. There's a lot of education that goes on when people go on to trials. And this idea that you have increased medical attention from trial staff and physicians is real. Patients do better when they're on clinical trials because they're being watched more closely. And they're being watched more closely because the physician who is running this trial, if something happens to a patient, they have to decide whether they think it's related to the treatment or not. And they have to dig in a little deeper, and they have to pay more attention. And most of the time when people and physicians are paying more attention to their patients, they end up giving them better care. The risks, it does take additional time. And, this can be very precious time to be taken. There's a possibility of known and unknown side effects. Sometimes there could be additional costs, like some of the biggest costs many times is having to pay for parking that you have to show up to, a particular institution. And sometimes the parking is quite expensive, and travel to and from. And then the idea that, there is potential risks for protected personal information. It's certainly something that patients are consented for, to make sure that this information could be shared, especially with the sponsor or potentially a pharmaceutical partner, so that they understand what's actually happening to the patients. You guys, I'm sure you know this well. There's a huge need for new therapies in glioblastoma. When you consider there's 8 billion people on the planet, we end up having around 240,000 people who are diagnosed each year. Of the primary brain tumors, glioblastoma is essentially half. And then, we've done really not a great job. And it's really embarrassing to say that there's only been 3 drugs or 3 approaches that have been approved over the last 40 years for glioblastoma. So GBM Agile was set up to develop these late stage trials, these kind of phase 2, phase 3 trials, with the goal of having it be a registration trial. And the first thing that happened is, there was an assembly of a bunch of experts from around the world to develop the protocol. There was a lot of collaboration with the various regulators from not only the US, but other countries. And then to try to create this environment where it would limit the downtime in between trials, and also, to save time between this phase 2 to phase 3 transition that occurs. Because mostly what would happen is you'd have a phase 2 study, you'd shut it down, you wait for the results, and then you'd say, okay, I'm going to go, and you go to the phase 3 study. So this is trying to get rid of that white space that occurs between these studies. And GCAR was developed to be initially the sponsor for GBM Agile. And it was developed in 2017. It is really the sponsor for the trial. So what you kind of have here is you have GCAR in the middle that is running the trial. You have the various groups that have been involved, including patient advocacy groups, pharmaceutical partners, regulatory partners, and then the execution of the trial, which involves CROs, vendors, and then the various sites. And so GCAR sits right smack in the middle of that. It's kind of the honest broker, and it's really interesting as we were dealing with regulatory groups that because we were kind of that honest broker in that setting, that we were given a lot more flexibility than many of the pharmaceutical partners would, because we're evaluating many therapies over time, and we'll get a better sense of that. So, as I said, it's a phase 2/3 trial. It's something called an adaptive platform trial that I'm going to talk a little bit about. Again, evaluating multiple regimens, newly diagnosed and recurrent, and it's a global study. And AGILE is an acronym. So this is what it stands for, is adaptive, and that has to do with the design and the ability to adjust based upon results as they're coming in real time. It's global, which, it's in multiple countries across the world. Innovative, well, at least we thought it was innovative. Hopefully you guys do too. A unique platform to make drug development cheaper, faster, fewer patients. And really, that's a really interesting feature of this, because having it be cheaper with less patients and all that means we get answers quicker, and more pharmaceutical partners are able to come on because the price has been lowered by at least a third, which allows really more shots on goal, which is what we need for our patients. And then, since you're going to see that this is a longitudinal trial, that arms come and go, that we are gathering a bunch of data, and that data can be used as a learning environment where there's perpetual learning that could be taking place, and really the type of learning that takes place is broad. Could be how we evaluate images, how we evaluate patients in different ways, and the tissues that come with it. So looking at kind of exploratory evaluations of a therapy looks like, well, it didn't do well overall, but we could dig in a little bit to see if there are subgroups that really seem to benefit. So again, the goal of GBM Agile, which this will make sense a little bit more as we get into it, but it's to rapidly identify experimental therapies that improve overall survival. That's the endpoint, getting people to live longer, and confirm efficacious experimental therapies, associated biomarker signatures. I'll get into what a biomarker signature is, to support the new drug approvals. So this is a schema of GBM Agile. And what you have from the very beginning, and actually these are somewhat temporally appropriate, where we had our first arm. Each of these areas here indicate an arm, and these are all the experimental arms that are either ongoing in Agile or have been in Agile. And then there's this control arm, and the control arm continues over time. So the nice thing about that is you can have a relatively limited number of patients who have to go into the control arm, because you get all of this power that's generated based upon that. And that allows us to see these signals associated with these therapies easier. And so we don't have to enroll as many patients into these therapies either. One of the interesting that happens is, as we're going through, and we've taken GBM Agile, and we have these two stages within GBM Agile. That's different than the phase. These are stages within the study. And basically, there are three populations that are being evaluated. We call these populations subtypes. So the subtypes means that you have recurrent GBM, you have newly diagnosed methylated and unmethylated. And these are subtypes because they have different outcomes. The recurrent GBM is shorter, unmethylated shorter, methylated longer. And so these patients come into the study and they get randomized. And based on the number of arms that are there during this first stage, as they come in, they'll be randomized to one of these arms. And in general, if we have 20% of the patients going to control and 80% going to other therapies, that's a much better ratio. That not only is that ratio favorable, but monthly, as the study is going on, they look at the data. The central algorithm that is being run evaluates the data, and for any person coming in, let's say it's a newly diagnosed methylated patient coming in, they evaluate all of these therapies, and the ones that are performing the best gets a higher likelihood of having it being randomized into it. So this is the adaptive randomization, increasing the likelihood that as a patient comes in, that they're getting randomized to the drug that is performing the best for them. Again, these are randomization probabilities that are being adjusted. This goes on throughout the study. And during this first stage, if we just assume this is stage one, as we're going through here, it's 200 patients that are being evaluated. And it could be, a mix of newly diagnosed and recurrent. But at the point of 50 patients that are being enrolled for a particular arm, we start evaluating for futility. And that means if a drug is not doing well in all of these groups here, then it's going to stop. That'll be done. It didn't work. And we find out very early that it was not beneficial, and we move on. The second option, and that's from 50 patients to 200 patients, is that we achieve something called graduation. And that means that as the algorithm is looking at things every month, it identifies that there maybe is one of these subgroups or all of them, or that's what these different signatures are. Like it could be methylated alone, unmethylated alone, recurrent alone, or it could be all patients that are being evaluated to see if they're showing a benefit across the board, or all newly diagnosed. And if they are showing that benefit, they graduate and go to the next stage. And that next stage enrolls 50 patients, a higher percentage because it's doing well. So we want more patients to get on. And then at the end of that 50th patient of that second stage, we wait a year and then we report it out. And if there's success, the FDA said that's going to allow us to take that forward for a market approval. So the final thing that can happen is that as we're enrolling patients, we reach that maximum number, that 200. And, sometimes there could be still some benefit, but it's not a very big benefit. We're looking for something called a hazard ratio of around 0.65, which means a 35% improvement in the outcome of the patients. That's the effect size that we're looking for. So if we see something that's 20% or 10%, it's not going to be picked up here. You're going to go and have your maximum enrollment at 200. Then you're going to let the data mature. And what then will probably happen is the company will need to look at that data and say, all right, if I have an effect size of 20%, am I still going to want to go forward and try to develop this drug? Because 20% improvement is not going to be that big of an impact. So we got to be careful about that. So those are the types of things that will go into the decision making. So you have early futility, you have a graduation, and then you have the maximum enrollment where people will be, and the arms will be considered for therapy in the future based upon the results of those data. So a little more about the experimental arms in GBM Agile is that the experimental arms change over time. You got that sense here. They're coming in over time, and there's new ones that are coming in. Patients on all arms have the same base visits and assessments so that there's consistency in how they're being evaluated. The patients on each experimental arm could have arm-specific visits and assessment, but those are, if there's unique toxicities or other things like that that need to be evaluated. And the drugs could be administered through a number of routes. We've had infusions, intramuscular injections, and oral injections. These are the subtypes again. And what I showed you is that there is a group of recurrent disease, newly diagnosed, and newly diagnosed unmethylated, and methylated. And these subtypes are the unique, when a unique patient comes in, they're going to fit into one of these subtypes. And they're going to be randomized and adaptively randomized and then go forward. But when the arm is being evaluated, that's going to happen with these signatures. And these signatures include the 3 subtypes, but they also include, as I said, all newly diagnosed or all patients. And this is where kind of additional savings and power comes from this, so that we identify what the population is that's doing the best. And that's the one that, of course, moves forward if graduating and for a new drug application. So this is kind of complex. You have multiple different types of therapies, and a patient comes in, you can't actually tell them a lot about each of these therapies. It's a little bit hard, and the patients don't get to choose the therapies. So the patient comes in and they're introduced to it. They will sign a screening consent, and they have to meet all the eligibility across all the arms coming in, and that's what these screening procedures do. And then they get randomized. And when they're randomized, they get placed into one of the treatment arms. And then at that point, they sign the consent. There's more data provided about that, and they start within 3 days afterwards. Again, this is in the setting of newly diagnosed and recurrent. And so that's the process that the patients have used, and that has been working out well. I talked earlier before about this idea of a learning environment, and we really are collecting a lot of data on patients, and there's a lot of opportunity for us to learn. We're always struggling with issues around imaging, and we hope that when images are being stored, we're storing them all centrally so that they would be accessible for researchers going forward. As I said, there's tissues on every single patient and blood samples on every single patient. So the idea that we could get a better sense of subtypes that may be benefiting, or just a better understanding of the disease and the biology around it. And then, with that will hopefully come new hypotheses for biomarker development, and that will help in selecting new drugs and considering new drugs for the future. So these are the main pillars of GBM Agile. Again, we feel it's very patient-centric because we're focusing on subtypes, and that patients can be randomized to several types of investigational agents. The fewer patients in the end receive standard of care. And again, strong engagement of patient organizations, but also this idea of adaptive randomization occurring. So the increased likelihood that that patient's going to receive the best therapy for them. This work is different. The statistics that are being used are different. These are these Bayesian statistics, which allow us to look very frequently at the data, or at least the algorithm to look frequently at the data. And it could do that because they run a series of simulations to see what are all the possible outcomes that can occur with this particular therapy. And with all those outcomes, they can go ahead and then make a decision about, again, whether it's graduating, all these things, by looking at the data right in time. If you've ever seen a trial that has a p-value of 0.000001, that means they enrolled too many patients to get an answer. Here, we're enrolling till we hit the mark, and then we don't need to enroll anymore, and then we allow the data to mature, and then we could move forward. The rapid timelines is just so that we can really move forward with these randomized clinical trials as quickly as possible. Our recruitment rate is 3 to 4 times faster than the traditional ones. To date, we've already have 1,900 patients that have been screened for the trial. And as we said, for the learning environment, that generates a large, highly annotated longitudinal database for other advances. We create the infrastructure here to be utilized by other partners coming in. So again, fewer patients needed, at least a third of the cost, which really brings in new partners in this. And I'll say, just to go back here, most of these partners would not be able to enter the trial based on standard costs. If we use standard cost and didn't use Bayesian statistics, instead of 200 patients in an arm being evaluated, you'd have to have 400, 500. Just as an example, for those who may remember, BMS did an evaluation on nivolumab, and they looked at recurrent, newly diagnosed methylated and unmethylated, and it was around 1,500 patients overall that were enrolled, and half of them were in the controls. If you simply multiply that by $150,000, you end up with around $220 million. That's a huge, huge cost. And many of the people who are small biotechs coming up with novel approaches, novel ideas, would not be able to afford that, but they were able to get in and have their drugs be evaluated within GBM Agile. And then, not only is it that it's cheaper and fewer patients, but when we do get and see a benefit, it's going to be in multiple settings. So the idea that regulatory groups would be able to, across the world, be able to move forward and to allow for market approvals in those settings, that's the entire goal. It's not just to have the United States, Canada, Europe. It's like as broad as possible that we could roll out effective therapies, because we know how important this is. So other things I think that we've touched on already, but I'll just say again, there's multiple investigational options. The design has been informed by patients and advocates. The idea of accelerated outcomes, and really this is a disease-centric, it's like you're throwing, there's drugs that we're being able to evaluate here, but it's all about glioblastoma, and it's all being compared kind of in the same environment. And that leads to a lot of statistical savings as well as financial savings. And then the idea that this could, an effective therapy here would move into standard of care. The nice thing that happens is once we begin to change the standard of care, that gets integrated right into GBM Agile. And then you could imagine that there could be combinations that would come right out of that. Adding immune therapies onto whatever it was, adding other things that make sense that could be used in combination. And all of that would be just allows us to keep moving faster instead of these very slow changes that can occur, take 15 years till we find another combination, something like that. This is just trying to accelerate that process. Once we see advances, that we would be able to accelerate. So I think, that you're an informed group, and thank you for joining. And I think this, how do you find information about trials? Of course, ask your physician, but you could go on the clinicaltrials.gov and you could see, not only information about the trial, but where it's being run. If you went on clinicaltrials.gov and you looked at where the different sites are for GBM Agile, you would see what those different sites are. You'd see which countries they're located in. And then also, folks like Al and other people who are so effective with patient advocacy and making sure they know about trials. It's always just a great group. So I'm going to stop here and see if you guys have any questions that you might have, and see if Al, you or Vanessa have any questions.
Yeah. Thank you so much for that presentation. That was great. And actually, when you were showing the 5 pillars of GBM Agile, the first was patient-centric, but as you went through the other ones as well, they're all patient-centric
Yeah.
because the design is just really, really gonna get us to answers faster. So just, I think you mentioned, how many sites are there globally?
Yeah, there are 60 sites. There's
60
around,
sites. Okay.
in North America, around 43, I think, and then in Europe, 17 to 20, and then in Australia.
Okay. And I think I asked Rachel from TCAR this, but so this is probably the largest GBM trial in the world right now. Would that be fair to say?
Probably now, based on the 1900 screened, I would say yes.
Okay. And one of the first questions that came into the chat is, why are there so few patients enrolling in clinical trials? And
Yeah,
you can have your
yeah.
own thoughts on that. You know, I always look to the inclusion exclusion criteria.
I think so too. I think that's a big, I mean, it's hard. Well, first what we're trying to do, I mean, um, just clinical care of our patients and trying to see things gets noisy sometimes, right? And so this is a way you kind of want to make sure that the patients are on drug long enough, that the patients have the opportunity to respond to the therapy. And so the inclusion exclusion criteria kind of creates homogeneity. There is definitely a selection for better functioning patients, but that also should allow us to kind of optimally see like what the effect of is a drug. If somebody got the drug for a week, they're not going to be able to tell what's going on. It doesn't help everything. So these are kind of our shots at getting it right. So then we could get it to everybody. And I think that part is hard. I think there's a lot of patients that are being taken care of out in the community. And there is not necessarily accessibility to these trials out in the community. And that's also hard. You know, I wish we were in a position where we would have so many arms because we could take on the capacity. If we had, like 7, 8, 10 arms, anything like that, we can actually be able to do that. But we are enrolling only a relatively small number of patients within each arm. And this is, I wish it was a mechanism to like have people, just everybody be on clinical trials. It's like, that's what we should be doing. That happens in pediatrics a lot. So, that would be ideal. But I'll say the other thing is, and this is a kind of a cynical statement. I'm sorry for making it, but many places understand that they can generate revenue at their hospital by treating glioblastoma patients. Surgeries, radiation therapy, imaging, and they many times try to hang on to him. And I'm really, I think that's really difficult. And I think it slows down our ability to kind of move forward. When you look at the pediatric setting, no pediatric group hangs on to their patients. They send them immediately to whatever place where there's going to be a trial that's on, and really super kind of amazing approach with that. I wish it was different in adult glioblastoma, and I wish there was more incentives to make sure that patients got to centers of excellence.
Absolutely. I think that's fair to say. Specifically for GBM Agile, could somebody who was inoperable, either for the newly diagnosed arm or for the recurrent arm, could they qualify for Agile?
As long as they had a biopsy, one of the things we need tissue because we're separating them into these groups, right? Methylated, unmethylated. So we need enough tissue. It's not a lot of tissue, but we need enough tissue. And we got to know that it's an IDH wild type glioblastoma. So in order to, clearly have that diagnosis, we need some tissue. And then to create these subtypes, we need tissue as well. But otherwise, yes, could be large tumors, small tumors, whatever.
Okay. And in either group, newly diagnosed or recurrent, is concurrent use of Optune while you're on trial allowed?
It's a really great question. And it's something that we talked about a lot. And we talked about the FDA with it a lot. There's a couple things I think that happened with Optune that One is the level of compliance that patients have. It's mixed. And if you have mixed compliance, you're going to have mixed outcomes, and that's going to be a little bit hard. The other thing that happened is there was uh, a study that was done that looked at, and they said, we'll allow Optune patients on. They would be randomized into kind of a separate Optune arm. They would have the clinical trial plus that. And then by the time they got randomized into that, a very small percentage of people actually took it, which is kind of like if you look at right now, even if you were to look at the number of people who are on Optune, and this is kind of worldwide, it's around 3,000 and something, right? That's a percentage. Maybe it's 10%, maybe it's less, I don't know. But that's concerning then, like who are the people that want to do it, and then how do we make that happen? I will say that we haven't had a problem enrolling patients not being on Optune. That has not been an issue. But I think if there was some way of figuring out that better, that would be good. But again, in the end, it doesn't appear like a high percentage of people end up going on it.
Okay. And then let's see. Okay, well, you mentioned just following up on Optune because some people do consider it standard of care now.
Yes.
So you're using, I'm assuming, temozolomide as your control arm for newly
Yeah,
diagnosed and lomustine at the recurrent phase.
that's correct.
If outside of this trial, if the standard of care does change, how does that impact the design of the trial and how the trial proceeds moving forward?
Yeah, I mean, so we were also careful about this with the FDA, and in particular about what do we do about this? Is this a concern? Can we say that this by some would not be considered the standard of care? And they said that that's not an issue. They were clear about that. That you'll still be able to identify other therapies that could be effective in that setting. It would be helpful. But again, how do we get to your question is, if we establish something that shows a benefit, then what is the standard of care, right? And how do we think about that? And I think, I mean, one of the really great things about Optune is like there isn't drug-drug interaction. And so, adding that to whatever else comes out would be an obvious and easy thing to do, because that would be kind of the first thing. Is there some toxicity, some drug-drug interaction that would be problematic? So that doesn't appear to be an issue, which is great, right? So I think it'll be okay. I think people will be okay with that in the end. Do you think there's some things that I mean, help me out if I'm not thinking it all through?
I have personal biases because my husband is on Optune, but
And it's great, right? I mean, yeah, and you
absolutely,
gotta want
yeah.
it. You gotta own it, you gotta be ready for it. And yeah,
He's got 98% usage, so yeah,
that's amazing.
we're all in on that. But we turned down, at the newly diagnosed stage, we turned down a phase 3 trial because it didn't allow Optune.
Yeah.
So I know you're not having any trouble with enrollment for GBM Agile, but on the patient side, it can be frustrating.
And I just wanted to add one
Yeah,
quick thing.
sure.
We have a large referral service for patients to get them into trials. And the question always comes up with the choice of Optune versus entering a phase 3 trial or any trial that doesn't allow Optune. And you're trading a small known benefit for an unknown benefit. If
Yeah.
people in a very difficult situation, it would be fantastic if you could even have like a small token arm where, as part of your control group, well, not the control group, but a small arm of your best treatment plus Optune, just as like an exploratory arm to see, and then if there's any results, increase it like you use your algorithm.
Yeah, and
It's
there was
a big
different.
problem for patients to make a decision like that.
Yeah, we've had some interactions with Optune to think about how we can integrate it, and in addition to other combinations with their approach. And it for different reasons it just didn't work out, but
Yeah.
I like the
Of
idea.
course,
I
it's an
like the
added
idea of it. I like
yeah.
the idea of evaluating it, and it's just It does add additional complexity, but that's we need to be able to adapt to as much complexity as is needed.
And you've already, I mean, GBM Agile is so much more complex than all our other trials. So you're definitely moving in the right direction. And similar to the question, if the standard of care changed, would the trial need to change? In the same sense, we've had big developments recently, with RANO 2.0 and your
Yeah.
assessment criteria for imaging. And so is the trial adaptive in that way, that if we understand more, we get better imaging modalities, and we change how we're assessing imaging? Is that
Yeah.
going to impact the trial?
Yeah, so this is an overall survival trial. And the thing we think about is, how do we make sure people stay on drug for a longer period of time to allow the drug to have its best opportunity to help a patient? And that could mean not taking them off too early. We think about things like pseudoprogression, and we don't know what therapies may be causing greater pseudoprogression. And so we have used modified RANO, which essentially much of that is now being implemented at RANO 2.0. We're going to, since we have done all of our evaluations with modified RANO, and it doesn't change too much, we're going to continue with that. But there's another part that's kind of interesting. You know how everybody, when they're developing their drug, they like the opportunity of having accelerated approval. You like the idea that, hey, if there's tumor shrinkage in the recurrent setting, I want to take advantage of that, and I want to have a fast track with this. So we are looking at, since if it's a 200 patient study, and you would say, well, if they're equally distributed, maybe 60, 70 patients would be needed for to evaluate in the recurrent setting for accelerated approval. And the way that the guidelines that they have them now, it was draft guidelines that they put forward, and they said, if you're going to do accelerated approval, you either need to have another trial kind of going on and almost be ready with it, or you could do it within the same trial. And the idea that we could do it within an evaluation within GBM Agile, in that setting, we would incorporate the response rate as RANO 2.0. And then that's the information that we would use that could then lead to, and what you would do is you'd enroll your, let's say 60 patients. And then as it's trying to, the data are trying to mature, you'd have an early readout on overall survival and the possibility of getting accelerated approval and then having that confirmed based upon the rest of the data that would come in. So we're looking at those kinds of things to help with that and to help move that forward. But, really, response assessment is really around in a survival study is, when do you take people off? And so that's the thing that we work on to try to make sure we extend it as much as possible.
Okay, well, that segues really nicely into my next question, which is, if someone progresses on trial, there is no crossover to another arm. They can't jump to another arm, correct? They're out of the trial.
Yeah, so we had this discussion with the FDA very early on, and they said no. And the reason wasn't a great reason, I don't think, because it was like, well, then they're going on another trial. You can't tell what which was impacting overall survival. And of course, they could just go on another trial somewhere else, right? And have that impact. So at least you would know within the same environment. So we're trying to, I think we're going to talk about re-evaluating that. It's something that, certainly our patients want, our investigators want. And so we want to see if that can be evaluated again. Now that we're, at 6 arms, hopefully having our 7th come in soon, that they'll have a sense that there's enough data there to help.
Okay, and digging in a little bit deeper, because I had this question and I've seen the question come through the chat. How do you pick which treatments enter the trial? Which ones you
Such a
start
good question.
on, which ones you've added?
Yeah, no, it's a really wonderful question. And we have an agent selection committee. Patrick Gwen co-chairs it with Ingo Mellinghoff, and we have a bunch of other folks that are on it. And you know, we have discussions with the company to make sure kind of would make sense within GBM Agile, just the way that they're doing it. We need to start like at the time of radiation. Some people maybe want to start after radiation or want to start at the time of surgery. And, you know, all the data that we have would not work for that. So it's, you know, kind of clarifying some of those things and if it would be appropriate. And then we have a presentation, and the presentation goes through preclinical data, all the existing clinical data, if there's a biomarker that they're interested and want to evaluate. And we try to make sure that they show kind of the imaging evidence of effect, all these things. And so we have a presentation, and then we have a discussion without them afterwards, and then we give the information back to them. Sometimes it's information indicating that we don't think you're ready yet. We think you need this level, you know, a couple more pieces of information that would really help us and solidify whether this is something that we think that we want. But I'm going to say something that may be a little controversial for those of us who think we know everything, because we don't. And I include myself in thinking sometimes I know everything, and I don't. We can see a therapy come up, and we can think that, oh, I don't like that mechanism of action. I don't like this. I don't like that. But this is really set up to be able to evaluate pretty much any drug and to be able to report out on it if it's not doing well with only 50 patients in. So you get this opportunity to kind of let the clinical experiment tell you whether the therapy is working or not. And sometimes I think that's very refreshing to have that rather than, you know, we're really good at neuro-oncology. We've had a lot of failures, so we're really good at identifying, like, oh, I would never treat with that or this or that, but you just never know. And sometimes you need to do the clinical experiment to know the answer.
Okay. And just thinking, you know, with the scope of the trial, when you have this many sites, I'm assuming there's still a big cost component. Like, how much does it cost to treat and track each patient in the trial?
Yeah.
And is that a cost barrier to smaller companies that may have good candidates, but maybe can't afford, you know, participation in a trial of this magnitude?
Yeah, and so far we've seen, you know, since we've lowered the cost quite a bit because of the number of patients and a number of things that kind of provide cost savings. Like I said, it's about a third of the regular cost. We've had more people come in. Like, I don't think, as I said, maybe four, and the, you know, or of those current, or the companies that have been in, four of them probably would not have been able to come in. Are there others that could come in at a potentially, if it was at a lower level? You know, I mean, for the most part, I'm having to learn a lot about venture capital by going through all of this, because you begin to see where people are willing to take risks and not. And when there's going to be a final answer at the end, they're willing to take more risks than if it's just going to be, I'm going to do a phase 2 study, we'll see what's happening. But if you go like, you get a yes, no out of this, then I think there's a tendency for people to be in, and especially if it's not as expensive as typical clinical trials. But it still costs money. It still costs money.
I was gonna say it still costs a lot of money.
Not free.
What do you think is the biggest cost component? What would you say?
Um,
I've seen it for other
I think
trials.
a mixture of, you know, CRO and site costs.
Okay.
Those are the two, you know, the per site costs and the CRO. Those are the most.
Okay. And so correct me if I'm wrong, there's been 6 investigational arms, is that right?
So there has been there's 6, some are ongoing. We've reported on one.
You reported on one. Okay, for the ones that are not ongoing, that stopped after the stage 1, do we expect readouts from those 2?
There should be, there's going to be one presented at ASCO.
Which one?
And I don't know if I could say.
Oh, it's okay if you can't say it. We had someone specifically in the chat asking about the paxilosib readout.
And then we have one that'll probably be at SNO, Society for Neuro-Oncology.
Okay.
Yeah.
So before the end of this year. Okay.
Yeah.
Okay. And then you have another one. So just on an annual basis, what do you expect kind of the tempo to be as far as arms, you know, hitting that 150 and
Yeah.
not making their signal and new ones being added in?
I think we're always looking to add new arms in, and we're ready to. It's just a matter of our company's ready. To some degree, do they have the funding? But that hasn't been kind of a major impediment. It's just been more, are they really ready? Do they have the data? We communicate with a lot of people that are just getting going in phase one to kind of give them a sense of like, what do you have to have available to be prepared to come into GBM Agile, so that they think about those things and that they're considering it. There's also, issues around dosing that could be not clear. I mean, it's really selecting a dose is not easy. There's again the traditional tolerability, but what's the best dose to use? This can be really, really difficult. And we could even answer those kinds of questions within GBM Agile. So for instance, you can have these two doses that are coming in and they're being evaluated, and the algorithm picks the one that's the best, and then you carry forward with it with a relatively smaller number of people. And then you have combinations. Combinations are like, that's what everybody wants, right? We really don't think that single agents are going to kind of do the trick. So it's about combinations. And with combinations, you put two in there and let's say you got a hit. And then the next question that should have been asked was, well, which one's contributing to what? Are they both contributing? Is it just one that's providing the predominant effect? And so we could build in these factorial evaluations, which would be single agent combination. And again, the two single agent ones would be dampened down with regard to the sample size. So it wouldn't be as expensive, but this is, normally what you'd have to do is you'd have to have, let's say, a 4-arm trial: control, single agent combination. And those can be really expensive. So we're trying to, again, bring all that down and come up with the Bayesian statistics that allow us to do things with smaller numbers of patients.
And that was going to be my next question, was about combination therapies, and that's what everybody wants. And do you envision at some point that we will get to combination therapy arms with Agile?
It's always the obvious combination now is kind of immune therapy. So throwing in a PD-1, PD-L1 antibody. There's a lot of interest in that. But the one that will probably drive most of the combinations is when we have a success, which hopefully will be soon, who knows, but we just want it as soon as possible. That's where combinations will probably move quicker. Because when you have a success, and it's kind of interesting, I mean, that hasn't really happened with temozolomide. We really haven't had a good combination with that. And again, that whole time frame, everything was very slow. We didn't move quickly to consider combinations. And then, the company runs out of its patent life and nobody's there to provide kind of the funding to be able to do combinations. So it's getting that right away makes all the sense in the world.
Okay, and this is slightly off topic, but because you brought up the checkpoint inhibitors, can you just talk for a second about the timing of checkpoint inhibitors and kind of your personal thoughts on checkpoint inhibitors in relation to, immunotherapy, surgery, or with Optune?
The checkpoint, I think we have just a real problem in the brain with very few T cells. And, just to kind of start back all over again, it's the reason melanoma and, other therapies like non-small cell lung cancer, squamous origin, where there's lots of mutations, lots of abnormal neoantigens, looks abnormal to our body actually responds to it and starts up a nice immune response, but it gets put to sleep by the PD-1, PD-L1 interaction. And so this is a single deficit that exists. Otherwise, if that wasn't there, our body would have taken care of it. It would have been no problem. So now we just use this antibody, we take over this single immunologic deficit, and now the T cells wake up, clones expand, and we wipe out the tumor. We're dealing with a setting with very few T cells. And so although we could talk about, neoadjuvant approaches, giving the drug, doing surgery, maybe trying to do our best to allow for the best opportunity for the checkpoint inhibitor to work, what we really need is an influx of T cells. We need something that's going to be an influx of T cells that's going to overcome the myeloid population, this is this tumor microenvironment. And in the end, we're going to end up having to deal with the PD-1, PD-L1 problem. Once we get that T cell population in and start that inflammatory response that becomes the immune checkpoint. So we have a lot to do. We have really three main things. Drive in T cells, get rid of the myeloid population, and be ready for the PD-1, PD-L1 interaction that's going to lead to our immune suppressive setting.
Okay, that brings up a brief question for me too. All the drugs right now are being looked at for overall survival. But if they did have, like they induced a immune response that was really exciting, that wouldn't accelerate them forward. I'm assuming those results though would encourage the funding to take them to a separate trial where they would investigate that, because that would maybe make it a prime candidate for a combination therapy.
I think we need to learn how to, our best response to inflammation in the brain is steroids, which is a real problem. And we need to learn some of about some of these other approaches, the IL-1 inhibitors, the IL-6 inhibitors. How can these best be used in a setting of really an activated inflammatory response? Because I feel like we end up not learning anything when we kind of pre-dose with steroids or when we give steroids soon afterwards, because we're just taking out a major part of the immune response. And it's really hard, and it's really frustrating to have that happen. And then you just, like you say, it's like you see something you think like, okay, there's something interesting. I'm seeing inflammation. I think I'm seeing a response. How do I capitalize on this? What can I do to do that? And the brain is still a really important organ, and it doesn't like inflammation in it. So it's hard. I don't have as much as I like investigating immune therapies. It's hard.
Yeah, we're getting close to the hour, but I do have a few more questions if you
Sure.
Okay, I'll try to wrap it up after the next few. We did get in a question, just to rewind back a little bit to the kind of the trial structure. So for the newly diagnosed arm, you have, TMZ as your control.
Yeah.
For the investigational arms, you're only testing the investigational drug, not together with TMZ?
No,
And then
it's a great
regardless
question.
of whether you're methylated or unmethylated?
No, it's a great question, and it's a mix.
Okay.
And some people, some of our partners have wanted to go in with temozolomide. Some of our partners are looking to replace temozolomide during the adjuvant setting. Some of our partners want to not use temozolomide during radiation, but during the adjuvant period. It's a mix, and it all has to do with the mechanism of action and the potential for interaction with temozolomide. So if these are drugs that have overlapping toxicities with temozolomide, that's going to be hard.
Mhm.
That's going to make it difficult. If you have a chemotherapy and you're adding a chemotherapy on top of temozolomide, you're going to get into trouble. So yeah, so those are, luckily we could pretty much do anything.
Okay. And then also, specifically for patients that, have done genomic testing, have gotten their NGS, and if they have specific markers, like their KRAS test identified that a PI3K inhibitor might be great for them.
Yeah.
They want to get the PI3K inhibitor in this
Yeah.
AGILE trial. So what do you say to patients, in that scenario? Should they go look for a different trial that's more targeted?
I think that I think it's been kind of interesting because we have a lot of preconceived notions about, well, I get my CARES report. It told me I should be able to use this and I should use it, but we actually don't know about what the best population is for those therapies. And it's we oftentimes are wrong. And that's why I think we ask the companies ahead of time, do you feel like you have a biomarker that's of interest? Because we can create 6 subtypes. If we have a biomarker, that one biomarker that they say, you know what, this is the biomarker that we're really interested in, that you could get adaptive randomization favoring all the patients going to that biomarker. And then if a patient comes in, they're more likely to get that therapy then. And so we want to be able to do that. Most of the companies don't know what their biomarker would be. Even if you have a PI3 kinase inhibitor, mTOR kinase inhibitor, they don't know that, PIK3CA mutation is the right mutation or makes the most sense. So it's a lot of it is still that we just don't know what the best match is for these biomarkers.
And I think, there's a growing consensus, I guess, that maybe we're going to need AI to get us there. Do you see AI playing a role at some point in, maybe incorporating registries or, what do you see as AI's role for GBM Agile or for kind of the trial?
Yeah, so first, Bayesian statistics are kind of like AI, and these simulations that are run as a way of creating data for it to adjust to and to learn on. In order to have to really have a huge impact, you've got to be able to take in gobs and gobs and gobs and gobs of data. We don't have readily available data. Now, GBM Agile is providing, could provide, let's say, 2,000 patients, whatever, and over the years more, but it'll all be still within that realm of no more than 10,000 patients. And I don't know if that's enough to be able to kind of do the kind of work that we want to see done. It might be, it might not be. It's probably the best setting to see it because, again, of the homogeneous population and to be able to identify whether there are changes. I think, so yeah, so we'll see. We'll see what happens with it. I wish that it would work really well and we'd have very quick answers. I think that would be great. But I also think, it's not just, I think, even MRI scans, I think there's a lot of AI that could be happening with that could create subtypes of tumors and look for responses with that. I think that kind of thing, because we have all the images, there's a lot of possibility with that. And those are dynamic, right? Those are over time. Whereas, you get your tumor sample, it's once, and then you treat, and then maybe you get another one, maybe you don't. But yeah, it'd be nice to have a more dynamic environment for that.
Okay, and last 2 questions. The first is, how long do you see GBM Agile running? Because it's an adaptive trial, it's continuous. It theoretically could run forever. So
it's built to be permanent. So we'll see.
And then my follow-up question is, how long do you think it's going to take to see a drug approval that will change the standard of care come out of GBM Agile?
Yeah. I'm hoping that no more than 10. That would, it's always hard, we always get frustrated, but it's still, no matter what, it's the fastest way for us to get a quick answer and to get it to as many people as possible, as quickly as possible. So even though I will be frustrated if we don't get it by 10, I will still believe that this is going to be the fastest way we could do this. Smallest number of patients, kind of a crisp evaluation, have a clear signal, and then get that to as many people as possible.
And just to put that into perspective, the normal drug approval time would be
Yeah, 10 years, something like that. I mean, we probably take, when you think of that, you're starting and you're doing a 2-3 essentially, and you're cutting out all the time in between there. I think we've estimated that enrollment, it shouldn't take more than 2 years, and then the final readout would be in 3 years. So that's a pretty short period of time. It typically would be 5 years, but if you add this phase 2, phase 3, that's more like 7 years.
And as patients and caregivers, it hurts to hear the 10 years.
Oh, yeah.
But I will say, and I heard this mentioned, referred to very early on when I first learned about GBM Agile, is that we're failing faster, which is a good thing,
Yep. And I'll say one other thing. It's like, sometimes I think we fail too fast with certain things because, this terminology, false negative, false positive, right? And to me, my biggest concern, what I have nightmares about is false negatives, because that means that we miss something that could be effective right now. And that's the worst. And that's why I think it's, we fail at the right time in GBM Agile. A lot of drugs, I can't tell you how many that we've been working on. And even over the last 2 to 3 years, there must be 4 or 5 different drugs that were stopped at near the end of phase 1 because they weren't going to be developed anymore. Some with pharmaceutical companies, some, that the biotechs couldn't get any additional funding. And it's because it's glioblastoma. And to think that one of those could have been an effective drug just is the worst feeling.
Okay, well, we're over time, but I just wanted to thank you so much for answering all of our questions. And oh wait, I got one more in just before we're about to close.
Oh, yeah.
And actually, it dovetails after what you just said. So why are there so many drugs that look good in the early phase, in the phase 1, phase 2, and then fail in the phase 3 for GBM?
Because they're not randomized. They are single-arm studies where the comparator that they use is not an accurate comparator. And when they do the phase 3 study and they randomize, they realize that they were just selecting for a patient population that was better, and they couldn't recognize that early on.
So hopefully trials like GBM Agile going forward will help us solve that problem.
Right.
Okay. Well, this has been great. Thank you so much for giving your time. This is a perfect way to kick off Brain Tumor Awareness Month. This is an important trial for patients and caregivers to know about. So thank you for sharing your expertise.
My pleasure. Thanks for having me, and thanks for all that you do. I mean, it's so important that you're increasing awareness of the population that you serve, and I know that they deeply appreciate it. I appreciate it too. Thank you.
Thank you. I just wanted to add that this trial design alone is a major breakthrough. We've been using the same trial designs for 100 years before this and never got anywhere. So this is a great step forward, and I think we're going to drastically speed up that search for the cure. The part I like about it best is in the past, like we watch how trials are going. We see like phase 3 trials going on for years and years, where we kind of know that it's not doing anything spectacular, that they keep enrolling patients just to finish it. Whereas
Yeah.
trial, that's not going to happen. As soon as you hit those 50 people, if it's not helping, you get rid of it and you don't continue to put people into something that's not going to work.
That's right.
So I love this trial. We have been sending lots of people to it.
Thank you.
Thank you
Appreciate
for
it.
tonight. You did a fantastic job.
Thanks. Okay, you guys take care.
Thanks all. Bye.